point of it is that there are large slits in between the endothelial cells and the fibers of the basal lamina in order to allow the cells to be filtered out and removed and phagocytosed. There are plenty of phagocytic cells with plenty of red blood cells inside of them, half digested, others not. Here the brownish pigment, if you look around, the brownish pigment in this cell, in the cytoplasm, if I move, that granulated brown pigment here is this hemosiderin. Again, it would be like brownish greenish if it was not that faded, but that's what I was talking about, the digested hemoglobin. There is another macrophage here. Basically, that's what you should remember. There are lots of macrophages which are there to remove the old red blood cells. We mentioned that there are specialized macrophages which present antigens. So not all macrophages present antigen. Those are the professional antigen-presenting cells which have the phagocytic function and also the capability to produce the MHC class II molecules and bind antigens. Here is again large macrophages with lots of debris in the cytoplasm. What else? That's it about the spleen. 80, slide 80 is the next one. And no, 34, sorry. 34, and we are going to the tonsils. I told you that the tonsils, okay, the tonsils are part of the secondary lymphoid organs, and they are localized in the oral cavity and the nasal cavity. There are 3 different tonsils that you should be able to recognize. One tonsil is localized in the dorsal part of the tongue. It's called the lingual. Okay. It is called the lingual tonsil. And there are 2 symmetrical tonsils in the back side of the oral cavity. They are called the palatine tonsil. They are hanging down from the soft palate. You all know that because I don't think there is anybody who never had a tonsillitis here. And then the third type of the tonsils is localized to the nasal cavity, to the back part of the nasal cavity, and that's called the pharyngeal tonsil. These structures contains many lymphoid follicles. They are partially encapsulated with connective tissue. It means that they don't have connective tissue capsule towards the oral cavity or the nasal cavity. Their main function is to filter antigens that reach the system through the air or through the oral cavity. So the antigens get into these tonsils, close to these tonsils, and I showed you that Dr. Fuller gave the oral cavity lecture. Dr. Woods probably showed you that the oral epithelium is very infiltrated at certain places with lymphoid cells. And the reason is that that's how the lymphoid cells actually meet the antigens. All right, what is this? This is just lymphoid tissue. No, this is... No, this is not. Yeah, this is just... Okay. Okay, we hope that because 30 side.
You have a good 30.
34 is so dried out that 34.
34, yeah, 34.
34 supposed to be the palatine tonsil. No?
No, you have 34.
Why not?
I heard
Yeah, that was the time.
It's on the label.
I'm gonna film my shoes.
Anybody has
Hi,
a
shoes.
34 that is not dried out?
Very
It's
nice.
on the label, here,
No,
34,
that's,
35.
no, it's something big, it cannot be, it's probably.
Too big.
It's where it's supposed to be.
Right?
Thank you.
Yay.
All right, oh, this is good. This must be good. So the tonsils can be recognized most easily based on the surface epithelium that covers them, and also some other structures. So we look at the palatine tonsil. If you ever looked at, did you ever look at somebody who had tonsillitis, and the doctors recognize it by these little white spots on the surface of the palatine tonsil? So those white spots are the pus that is in the crypts that reach down deeply into the tonsil. And these crypts are just the surface epithelium invaginates into the tonsil, and these crypts develop to increase the surface where the lymphoid cells can meet the antigens in the oral cavity. Okay, so if you look at this tonsil here, you see that this is covered with the oral epithelium, that is the stratified squamous non-keratinizing epithelium, and you see the lymphoid tissue underneath.